This is supposed to be a tutorial talk, and it's a little bit different than the tutorials that I give normally. Usually it's what is compressive sensing or what is probabilistic data analysis or something like that. This year I decided to do something slightly different and give a few tips and insights that we've learned from assessing metagenomic computational tools. So the plan for today is to give you an idea of the context that we're going to be talking about metagenomics and this project called CAMI. And then we're going to talk about a few different things that you should consider when you either make your own tool and want to publish it and compare it to other methods, or do some sort of benchmarking study. In particular, utilizing standards, which are very important, choosing metrics, choosing visualizations, things along these lines. Okay, so let's start in the beginning. What is the context that we're talking about, metagenomics and CAMI? So I'm going to be talking about this particular use case of whole genome shotgun metagenomic sequencing, whereby you have some sort of environmental sample and you throw it on one of these sequencers, in this case an Illumina machine right here. You got all the genomes floating around. Those are not genomes, but it's the best thing that I could find that look like bacterial genomes. It shatters them, and then you output a whole bunch of reads, tens to thousands of gigabytes. There are many different microbes in these particular samples. And there are a few different things that you might want to do with this, studying all the microorganisms, bacteria, virus, and archaea present in that sample. There are a few basic things that you want to do first when analyzing a metagenomic sample. So you got all these raw reads sitting around. You might want to assemble them. And from that, extract longer contigs. And hopefully they will be long enough to then say something interesting biologically. So we got assembly right here. After you have these contigs, it's a community of microorganisms, so there's many different organisms in there. So what you might want to do is this thing called genome binning, which is partitioning up these contigs into putative single organism sets. So what I mean by that, you got a bin, I threw a couple contigs in there, hopefully that's from a single genome. Hopefully this is from a single genome, and I don't even know what this is down here, etc. So we got assembly, we got genome binning, you got taxonomic binning, which is skipping straight to figuring out which organisms all those contigs belong to. And then maybe you can try to assemble these a little bit better or scaffold them or something like that. Another, or the fourth major computational technique in metagenomics is this thing called taxonomic profiling. You don't care about assembly. What you care is who's present in the sample, what's the relative abundance of the organisms there. So, for example, at the phylum level, you get the relative abundance of the different organisms that are in your sample. By the way, feel free to interrupt me and ask questions at any point. I welcome it. Okay, so those are the sort of basic computational tools in metagenomics, but there are a couple different problems that you run into. First off, there's a lot of different tools out there to be able to accomplish these different tasks, hundreds of them, and a lot of what I call roll your own solution comparisons. So somebody comes out with a new tool and they make their own simulated data and they use their own metrics. And then they run some other tools and say, well, guess what? We're better than they are. So how do you really understand the actual state of the field? You need to do one of these large-scale benchmarking studies or pay more attention to the comparison section of the methods paper that you actually publish. So hopefully we'll be able to cover a couple of those today. In particular, what are the lessons that we learned from this CAMI benchmarking efforts? By the way, if metagenomics isn't your main area of interest, hopefully the different pointers that I'm going to be going over can still be applicable no matter what your field of application is. I've said CAMI a few times. What is CAMI? CAMI stands for Critical Assessment of Metagenome Interpretation, or Metagenomic Interpretation. It had its genesis back in a long time ago now. I think it was like 2007 or 8 at the Isaac Newton Institute at Cambridge. A bunch of us came together and decided that we need to try to bring together the community and solve this problem of hundreds of tools. How do you compare them all together? So we did this in a competition style format with a bunch of different participants scattered all over the world, and developed standardized sort of input-output formats so that we can use the outputs of the different tools together to be able to do a comparison. Things you can look at if you want to, and perform the standardized comparison. Try to use realistic simulated data here, and people are competing to rank to see who goes, is the best tool, and really assess the performance of these things. So this is what CAMI was, the sort of big picture idea, to be able to give biologists a roadmap for software selection. If you want to do taxonomic profiling, what tool do you want to use? There's 100 to choose from, so maybe you can use one of these benchmarking papers to guide your choice depending on your exact environment and what's important to you, et cetera. The other main goal is for people who do methods development and you actually write tools, how might you improve the tools that you are working with? By the way, CAMI is in its second round right now, so we are doing CAMI 2, another competition, and it's going on right now. If you're interested in checking that out, the link is here. I think these slides are also going to be posted somewhere eventually, so then you can grab links there. Okay, so that's CAMI in a nutshell. We are trying to take some of the lessons that we learned from CAMI and doing these large-scale comparisons, and let's talk about some of those now. So in particular, standards. Well, you can standardize a lot of things. Let's first off talking about data. Being able to utilize some open access standardized benchmarking data sets are really critical to be able to get an idea of the actual performance of tools across many different labs and different tools, et cetera. And in my mind, there are a few major things that you really want to make sure that you include in one of these data sets. You definitely want to include the raw data. It's amazing how many papers that you do, and they say they did, simulations and stuff like that, and you try to find the data, and you can't. It's not anywhere. You should also include the ground truth. Maybe they included the raw data, but they didn't actually tell you what was in it to do the simulation. And you should be able to make sure that things are machine readable and utilize identifiers for whatever it is you're talking about, so that it's easy for people later on down the road to figure out what exactly it is you are talking about. So we're going to go through each one of these and talk about it in a bit more detail. So, raw data sets. As I mentioned before, try not to roll your own solution. So here's a bunch of different tools for taxonomic profiling, and each one did their own simulations and their own metrics, and these tools came out on top, of course. And so attempt not to do this as much as possible. Utilize community-developed benchmarking sets. And there, in metagenomics in particular, there are sets that are like this. And I'm sure that if you go poking around enough in your particular application area, you'll find ones that are coming up in benchmarking studies pretty regularly or people utilize frequently. So try to use those things. In metagenomics, there's one, Alexander Sherba published it, so it's open access. You can go and you can download this thing if you want to, and work by other people as well, where they have nice, I'll stand right in your photo, other benchmarking data sets that are going to be open access. If you decide to roll your own solution, please, please, please try to make your pipeline reproducible and easy to use for other individuals down the road who want to do their own comparison and utilize the same sort of simulation that you did. In this metagenomic application, there's this thing called CAMI Sim, which is the simulator that we used for CAMI, a very inventive name, I know. But the whole basic idea here is it allows you to design a different simulation and then create that simulation, providing the raw data, as well as all the information that you would want, the ground truth, utilizing unique organism identifiers, these things that I'm going to get to in a bit. So spend some time before you decide to just simulate your own. Get an idea of if there are pipelines that are nice and reproducible that other people have already spent some time developing, you can steal and generate your simulated data sets. Ground truth. Obviously, this is what is in your simulation. Here's an example of a publication in Metagenomics. They benchmark a whole bunch of taxonomic assigner tools, and they provided the raw data sets, which is absolutely fantastic. And they didn't tell you what was in those raw data sets, which is kind of unfortunate. It makes it a lot more difficult to work with. You download those raw data sets, and you look at these FASTQ files, and you look at the sequence names, and it doesn't really tell you anything about what is actually in this. Other situations that might come into, Human Microbiome Project is one of the first ones that developed mock communities for different tool developers to test their tools on. And you can go to the website even today and access both the data sets and the ground truth. Sweet, you click on the link for the ground truth, and you get a PDF document. Oh, great, this is not machine readable. Now I'm gonna have to go through the laborious task of figuring out what they were talking about and converting these gDNA mass in grams to percentages for relative abundances and stuff like this. So in your ground truth, however you represent it, attempt to make it machine readable as well as actually available. There are a couple best practices that you can do here. So in particular, provide the mapping and metadata files. So pair up your raw data along with a text file or CSV file or something like that that tells you what was in it to begin with, as well as if you really want to make people's lives easier, in the raw data itself, say where these actual pieces of data originated from. So you might not be able to see it because the font size is so small, but here it tells you the reference was this particular organism, this particular part of the genome, et cetera. It'll make people's lives a lot easier, especially when the website goes down and these mapping metadata files are no longer available and you just have the raw data. You can still find it inside there. Any questions so far? Metagenomic stuff? I know probably only a small fraction of people here do metagenomics. Yeah?
I was wondering if horizontal gene transfer is a concern in some of choosing, like if this gene was always in this one bacteria, but it got horizontally transferred to another bacteria, and then it doesn't.
These can come from different organisms.
Yes. Yeah. Yeah. So horizontal gene transfer, as well as DNA from other organisms, viruses, et cetera, inside a metagenomic sample, it definitely complicates things and makes it difficult. So that was a couple of things that were actually measured in this CAMI competition. When you do assembly, making sure to pay attention to all the misassemblies of, well, maybe you stuck this gene in and it was actually, it doesn't belong in that one, it just got transferred and horizontal, that kind of thing. So it is definitely one of the metrics that you have to pay attention to. Yeah. Okay, what are these organism identifiers that I'm talking about? This happens a lot more frequently than it should. Don't ask me how to pronounce this, but I will try to slaughter it for you. Rautella ornithinolytica. Wait, was it Rautella ornitholytica? Oh, great. Which one's which? You Google both of these, you get hits for both of these. Great. Are they the same organism? Are they different organisms? How are you going to be able to tell the difference between these? In your simulated data, when you represented these in here, which one is the right name to use? If your mapping and metadata file that says what is in your simulation looks something like this, you're going to have a bad time. Well, maybe you're not going to have a bad time, but somewhere down the road when somebody wants to parse this thing, they're going to have a bad time because this is text. Well, it's nice and human readable. Misspellings are very common. Unique character sets, errant spaces, things along these lines, which make it very difficult to match up which organism is actually which organism. If you got one of these guys in here and your database uses that name instead, they're not going to match them up. And they got to worry about fuzzy matching and things along these lines. Ugh. To avoid all this, I'm recommending utilizing unique identifiers that are machine readable. So here's an example of something that might be a little bit better that uses tax IDs. Unambiguous, comes from a particular resource. You can go and look those up where they're from. Machine readable, no worries about using different UTF-8 versus whatever character set that you're doing. Because it's amazing that some different bacteria, they're named after their discoverers. Discoverers are all over the world. And so you got unique characters in here and stuff like that, and so they don't match up. Ugh, but in any case, try to use these. I would suggest utilizing a couple of different resources that are out there. Identifiers.org is one, which it's pretty amazing all the different identifiers that they have been able to assign to all sorts of bioentities. If you want to talk about a particular disease, they have an identifier to unambiguously identify that disease, or a symptom, or a drug, or a protein, or a pathway, or microRNA, or an organism, etc. And they're very standardized, machine readable, as well as they resolve to an actual location where you can get more information about that particular bioentity. How do they accomplish this? They utilize URIs, Uniform Resource Identifiers. And typically the things that you would try to put in your file instead are called queries. A query is a compact URI. What do these things look like? Well, here's an example. This identifies that from the NCBI source, I am talking about a tax ID with this particular ID. And so this identifiers.org and other resolver services will know, oh, if you want to go pick that out, they will go and look up at NCBI and unambiguously find where that thing is. Okay, maybe you don't work with lots of bacterial organisms. Maybe proteins are important to you. So here, UniPro-KB unambiguously identifies which protein it is that you were talking about. And you don't have to worry about nomenclature and whether you're using an abbreviation or one of the common names or synonyms and stuff along these lines. It uniquely identifies it. Maybe drugs. So here's a chemical compound as well. The really nice thing about these is that you have mapping services. So if UniPro-KB is not your jam and you want to use some other protein repository, there are mapping services that will convert one of these queries to another one. So like OXO is one. You have UMLS from the NCBI or NIH, and a couple others from RENCI. Oh, yeah, question.
Yeah, so I've even had this issue with NCBI where like different versions of the database they'll have like reclassified an organism as something else, so it changes. So does that also work for different versions of NCBI or whichever?
Yeah, so that is actually a choice that the particular places to which they resolve can make about provenance information and date information. I'm pretty sure with NCBI it always just goes with the most up to date. And so, if something did get reclassified and shuttled around, it's gonna reflect that kind of thing. However, the cool thing is since they give historical data dumps, it makes it a lot easier to look for it if you really want to talk about what the taxonomy looked like 2 years ago or something. You can go and then look for these exact matches in there.
I see.
Yeah.
So you could at least validate whether like, I said it was this and that doesn't match up with this benchmark, but actually it's just a different version, and you can check and make sure that's actually the case.
Yeah.
That used to be that.
Yeah, exactly. Yeah.
Do these identifiers allow for hierarchical structures? Because I'm thinking, for example, disease symptoms and
Absolutely.
terms where you always have very broad terms and more specific one and more specific one.
Absolutely. So ontology is basically what you're talking about. Wonderful. It's back. Yes, they do. And so, for example, taxonomy is hierarchical as well. And so each one of these guys actually corresponds to a node in there. And it might not be a leaf node, it can be higher up. As well as like human phenotype ontology, so for symptoms and stuff like this. The entire ontology is represented with these different numbers, sometimes why they're very long. But it does get you that individual nodes in the hierarchy, and then you can go resolve to the actual provider of the information to, for example, step one higher up in the ontology if you wanted to as well. Excellent. Standards, standard identifiers, standardized formats. It is a not so secret fact computational biology has an issue with formats. You know, what percentage of our time is devoted to just converting from one format to another? And so it's a great time for a relevant XKCD. Situation, there are 14 competing standards. 14? Ridiculous. We need to develop a new standard that covers everybody's use case. Later, situation, there are 15 competing standards. Yes. Yeah, curiously though, oftentimes there are no standards, right? Somebody writes a tool, they give an output format, and they tell you at the landing page of GitHub basically what it does. That's not a format specification. Right? It doesn't exactly cover all the use cases and tell you what data types to use and how long the fields should be and everything along these lines. So oftentimes there are no standards. And so, as an example, in taxonomic profiling, which you'll recall is where you take these reads and figure out the presence and relative abundance of organisms in a sample, it turned out there were no standards a number of years ago. However, if you look at a couple of different popular tools, and you can take a look at what sort of output formats they were using. So here, Metaflan, it was using the standard human readable ones. Okay, yeah, we already see a couple issues that might arise from that. Oh, Kraken. Hey, look, I got tax IDs. This is nice. But they try to do the hierarchy of these things vertically instead of horizontally here. Let's see if we might not be able to take bits and pieces from each one of these and develop a standard. So Cami developed an actual standard. So it's this YAML file, yet another markup language, that will tell you exactly what fields are optional and required and what sort of things can go in there, et cetera, and it's versioned. That way, as this gets updated, you are able to keep track of the change of these particular formats here. If at all possible, try to utilize some sort of standardized format in whatever output of your tool that you're developing is. If you don't, here's a hint. Look at like the most popular tools, try to mimic their format. Because people have doubtless created pipelines for the popular tools. And if you're outputting it in at least a similar format, it would be easier for them to plug your brand new shiny method in as well. Okay, metrics. Metrics that you choose to do your benchmarking study, choose carefully, choose liberally, and choose before you actually do the entire analysis. That way you don't bias yourself to picking the best metrics that show the performance of your tool. So in the taxonomic profiling example that I'm talking about, the sort of sensitivity, specificities, kinds of true positive, false negative kinds of things apply. But you really want to keep in mind as well the domain-specific metrics. So what do I mean domain-specific? Consider what other people have used, but especially consult with different experts in the field, because the particular metric that you think about as a CS student or somebody who's doing machine learning or more computationally minded might not actually be the metric that biologists really care about. Because you're worried about sensitivity, specificity, and they're worried about something completely different. Maybe how well did you get the organism abundances correct? For example, in this one, if you look at the output at the species level for Metaflan, where each one of these blue dots represent a species, you'll notice that they have no false positives. Oh, this is great. No false positives. Your specificity, wonderful. However, a biologist might look at this and say, well, this is not so good because your predicted relative abundance is way off from your true relative abundance. So we might need a metric that captures this a little bit better. So for example, you might want an L1 norm, which basically adds up the length of all these little blue lines right here instead. So keep in mind that end users or biologists might consider metrics a whole class of metrics that are different than the ones that you are utilizing. As another example, there's this other metric called unifrac, which is important in this particular area, where you take one sample, let's call it the blue sample. You got yourself a taxonomic tree right here. Your tool assigned a third of your sample at this node, and this node. And the yellow tool assigned a third of your sample here, here, and here. And basically, it tries to figure out how close these two tool outputs are by figuring out the most efficient way to be able to get one of these guys to overlap on top of the other, which, unless you had consulted with an expert in the field, might not be the first thing that you think about when you're trying to think of a metric to analyze the performance of a taxonomic profiler, or whatever method it is that you're doing. Is that a hand or a hand? Okay, cool. Yeah?
Do people use already EMG or not?
Yes. Yes.
They start using it.
They have started using it, yes. Yeah. Yeah, we have a new one out called Striped Unifrac out with Rob Knight's group. So basically they have incorporated it into QIIME 2. So it's...
But there is also us. We try to introduce it, which is interesting. Because, for example, what's the PPV specificity?
Mm-hmm.
There's a good, but if you have populations, in other words, sets with
Yeah.
frequencies, then what will be specificity?
Mm.
What will be PPV? And there is an element we kind of try.
Yeah, yeah. Yeah, definitely.
It's an interesting question. Yeah.
Definitely. And lastly, in the last about 4 minutes, visualizations. So really try to think what it is you're attempting to communicate to your audience when you do one of these benchmarking studies or you publish your own tool and want to showcase its performance. Are you trying to represent a cause and effect or a trend? Might consider a certain kind of graph, like a line graph. If instead you're trying to show composition, how parts relate to the whole, you'll definitely want to use something that looks more like a pie chart. If instead if you have different distributions and you're going to compare these together, might consider a violin plot, etc. So spend some time really thinking, what is the story that I'm trying to tell, and what's the appropriate visualizations to use? As a fantastic resource, you can consider this book. It's a classic visual display of quantitative information that contains a whole wealth of information about helping you pick and make good figures and graphics. A couple different takeaway messages from a source such as this one is attempt to ease reader communication. Strive for simplicity if at all possible. What does that mean? Maximize the ink to information ratio. So you can try to use little ink to represent lots of different information so that visually it remains simple. And then iterate and revise often on these things. So let me give you a kind of an example of this. So we're talking about taxonomic profiling. So in this case, in the CAMI competition, we had multiple versions of 10 different tools, 5 different metrics that we decided to utilize over 3 different classes of simulations, and you can do this comparison at 6 different taxonomic ranks. Ooh, great. Multiply all these numbers together, plus and the number of versions here. How are we going to try to visualize this information? Some people, I stole this from a publication that will remain anonymous because I'm picking on them, choose as figure 1 of their publication to be this nice big old table. So, quick, tell me which tool has better false positives? Yeah, exactly. You're gonna have to sit here and stare and try to figure out, and these are all abbreviations here, so, well, some of them, but what's, okay. Tabular information, it's particularly great if you do it in a CSV file because you mean for somebody to have a computer parse it, but for humans, not so much. So as a first approach, trying to ease the reader interpretation, you might say, well, let's just use a line graph, right? Because we got a bunch of different taxonomic ranks. We can put it here on the x-axis. We can put one of our metrics over here, and then all our different tools there. Marginally better than a table. But remember, the story that we're trying to tell here is not a cause and effect or a trend. So maybe a line graph isn't the best, and it's still not too simple because telling this color from that color, not going to be easy, right? So we might need to try something else. So they spend a lot of time thinking and looking at different graphics. You go to this visual display of quantitative information and you stumble upon, hey, radar plots. Oh, maybe this will be better. Here we can show different taxonomic ranks and all the different tools over here, as well as all the metrics. Definitely maximizing information to ink ratio here, but reader interpretation, still not the best. It's a little bit hard to see, well, this tool has the best precision, and oh, is tied for this one and this one across taxonomic ranks. So it takes a while to really understand. So we go back, we try to figure out what are we really trying to communicate? What's the story that we want to tell about these different tools? So, striving for simplicity, we revise again, and we get a plot that looks like this. Radar again. Now I've reduced it down to 2 metrics, and we can see, oh, something really striking happens. All this red surface area disappears as you move from family to genus rank. That's the recall. Oh, the recall just takes a nosedive for all tools when you go from family to genus. That is a nice little kernel, nugget, piece of your story that you can impactfully tell right here. And you might not be able to see just by looking at a table. Yeah. 30 minutes is a short time. Okay. To give you an example of our attempt to put all these things together, OPAL, which is a way to assess taxonomic metagenomic profilers, utilizes standardized input and outputs, community-derived metrics, and it results in interactive visualizations. So we can use CamiSim, which I talked about earlier, to do your simulation pipeline. Gives you your simulations. You stick it into different tools. They give you outputs. You combine it up with the ground truth that you did, and it'll stick out for you machine-readable tables, human-readable visualizations, as well as rankings. And you get pretty stuff that looks along these lines. So, for example, if you were to do this, and if metagenomics is your thing, you might want to utilize this guy. There we go. Nope, there we don't go. Apparently I can't switch windows. Okay, well, in any case, going to this particular website, it's pretty cool because it lets you choose different metrics and which metrics are important to you and change the rankings and things along these lines if it's kind of interesting. But the whole point of this talk is trying to be a tutorial about tips, not so much focusing on this particular tool. So, putting it all together, when benchmarking, utilize community-derived data sets. If you do roll your own, make sure to include all the relevant pieces, as well as the analysis results. I actually distinctly remember in one paper, they didn't actually provide the values of the accuracies, and I had to go back like with a ruler on their figure to try to recapitulate what exactly these values were, because they weren't answering any of my emails. Try to provide the analysis results. Employ standards or mimic popular tools, carefully choosing your metrics, and spending a lot of time on your figures. Lastly, in the -1 minute, shameless plug, I'm recruiting, so I recently moved to Penn State, so I have a lab to start up over there. If you're interested in metagenomics, computational biology algorithm development, and in moving from a math department, so mathematical methods, trying to prove something about a method, feel free to come talk to me about different available positions in CS and bio at Penn State. So I'll be happy to take questions at this point. Thanks for your attention.